In haploid culture, second type is the gynogenesis, in which female ovary or ovules are used for the haploid culturing. In this development is in which the embryo contains only the maternal chromosomes due to activation of an egg by the sperm that degenerates without fusing with the egg nucleus. So regeneration rates are quite low, and usually only one or few embryos that develop through the process of gynogenesis. This is basically used when anthrogenesis protocol fail in some species. However, this process has very low yield that lack any developed protocols, and production of diploids is very low. In ovary culture, basically the unopened, mature, unfertilized flower bud is used. By histological study, appropriate stage of ovary is determined, because here the stage of ovary is very important. So you have to do the histological study before using it for the ovary culture. Proper sterilization is mandatory in all cases of tissue culture. So there are two basic processes which are used for the ovary culture. First is the induction process, and second one is the regeneration process. In both cases, you have to incubate for 16 hours, and after 2 weeks, haploid plant lets grow. So haploid production is very low. In this case, 1 to 5% is the success rate. So some cases there is no need to be sterilized. Two methods are used. First is the filter paper support system. Second is the vermiculate support technique. Haploid production of onion and sugar beet has been done through the ovary culture. General gynogenesis procedure, as I have discussed in the androgenesis, there is also some general procedure which you will use. First of all, you have to use unfertilized ovary or ovule. Then explant is collected several days before from the anthesis, and then sterilization is done according to the species, that which species you are using. According to that species, you have to use the optimized protocol for the sterilization. Then during removal of ovule from the ovary, care must be taken, as I have discussed previously in case of androgenesis, that if there is any injury in the anther, pollen or ovule, then the process of androgenesis or gynogenesis cannot proceed further. So other factors which basically affect the process of gynogenesis, first of all is the genotype, that which type of genotype you are using, then stage of microspore development, then media, which kind of media you are using, either that media is suitable for that particular type of genotype or not, and others all other factors, temperature and light. As I have mentioned in all my classes, that first of all you have to optimize the conditions. That conditions also include the temperature and light condition. That conditions also need the media and the sterilization conditions. So if these all things are optimized, only then you have a successful tissue culturing.